please.
Hi, my name is Emily. I'm a sophomore. And over the summer, I had the opportunity to research with Dr. Baker, and we looked at the Ebola virus secreted glycoprotein and how that enhanced the maturation of dendritic cells. So this is the Ebola virus, and many people are familiar with the Ebola virus because of the huge outbreak that happened in Africa earlier in the decade. But Ebola has actually been discovered since the 70s, and so the reason why it became so popular is because of the outbreak in Africa. And so people started looking at its genome to figure out why is this virus so deadly. And when you look at the genome of the Ebola virus, it only consists of 8 proteins. So the question is how is something that's so small and only consists of 8 proteins so deadly and can kill thousands and thousands of people? And so the best way to answer this question is to start looking at each of these proteins and decide what do these proteins do. So the protein we looked at was sGP, and the reason we looked at sGP was, one, because there's not been a lot of research done on sGP, so it was kind of new, and second, whenever you look at the patient's bloodstream of someone who has been infected with the Ebola virus, there is an abundance of this sGP present. So why would the Ebola virus secrete all this sGP if there really was no purpose? And we looked at how sGP affected dendritic cells. So dendritic cells are antigen-presenting cells, and they're the first line of defense whenever an invader has entered the human body. And so you first have these immature dendritic cells that kind of survey their surroundings, looking for foreign invaders. And then as soon as they sense one of these invaders, they mature and they start presenting these antigens, which T cells and other immune system cells can then bind to start fighting off this infection. And so our question was, how does sGP affect these dendritic cells? And so our first thoughts based on prior research was, whenever there's sGP affecting these dendritic cells, they're just going to stop functioning. They're not going to call these other immune system cells to it, and therefore the virus can't be fought off. And so our original hypothesis was that dendritic cell function would decrease whenever sGP was present. And so to look at this, we first started with a precursor cell and looked at how sGP affected the differentiation from these precursor cells to immature dendritic cells. So to do that, we had a differentiation cocktail, and then we added or we didn't add sGP. And so we did 2 experiments to test and see how dendritic cells were affected. And so one was an ELISA, and ELISA basically you look for certain cytokines that have been present in the supernatant of these dendritic cells. And so we specifically looked for IL-12. So IL-12 is a pro-inflammatory cytokine, which means whenever this is being secreted by the dendritic cells, there's an infection and it needs these other cells to come fight it. And so when we ran that on our differentiation experiment, we found that the IL-12 concentration between these precursor cells, there really was no IL-12 being produced compared to those which this added TNF matures the dendritic cells. So that's when they're supposed to be producing this IL-12. So we saw that regardless of if sGP was added, these precursor cells weren't going to produce IL-12, which makes sense because they are precursor cells and they're not calling all these other cells to them. So from this, we concluded that sGP really has no effect on differentiation. And we also ran flow cytometry to determine how the presence of the markers CD83, CD86, CD54, and CCR7. And so basically what a flow cytometer does is whenever a dendritic cell has these antigens, we put a fluorescent antibody in and it will attach to these antigens and it'll be like a marker. And then you run it through the flow cytometer and it'll pick up all of those fluorescent antibodies and it'll produce a graph that looks something like this. And so this is how many cells are present, and this is how many of those markers are present on the cell. So as you shift to the right, there's going to be more markers present than if you're over on this side, and then how many cells have that many markers. And so the unshaded region is our unstained control, so no markers should be present there. And then our shaded region is our experimental group that do have the markers present. And so when you look at the data for our differentiation, this is CD83. And as you go down, sGP is added. So this at the top is the precursor cells, and this is when they've been added to be differentiated into the immature. And then sGP is added. You see that there really isn't a shift to the right. So there really isn't a increased presence of these markers, which would indicate maturation as you add sGP. And we kind of saw that in the rest of our markers as well, that there really wasn't a shift and there wasn't a greater presence in these markers on the differentiated dendritic cells. And so from that, we also concluded that sGP has no effect on the differentiation. And so the second step we looked at was these immature dendritic cells going to mature dendritic cells. And to do that, we looked at, we added a maturation cocktail and then added or didn't add sGP to them. And so the first thing we looked at was what would happen if we added sGP before we matured these dendritic cells with TNF. And this is what we saw. So again, we had the IL-12 concentration, and then we started with just the mature dendritic cells, so we didn't add any sGP, and then we started adding sGP little by little. So the 1 microgram per milliliter of sGP and the 0 microgram, so just mature dendritic cells, there really was no significant difference in the production of IL-12. But then once you added 10 micrograms per milliliter of sGP, there was a significant increase in the production of IL-12 in the supernatant of these dendritic cells, indicating that there's more IL-12 being produced, so it's alerting these other cells. So it's maturing and enhancing the maturation of these dendritic cells. And we also saw this in our flow cytometry data as you go down. So those are the mature dendritic cells that have had no sGP added, and as you move down, you see a greater presence of the markers on these dendritic cells, indicating that they have been matured, or their maturation has been enhanced. And then finally, we added sGP after we had matured the dendritic cells, so they were already mature when the sGP was added, and we saw the same trends with the IL-12, how when you just had it added 1 microgram per milliliter of sGP, the IL-12 concentration really wasn't significant to when it was just mature dendritic cells. And then when you added 10 micrograms per milliliter of sGP, there was this huge increase in IL-12 concentration. And from that, we concluded that sGP, even after the dendritic cells have been matured, it enhanced that maturation. And we saw this again in our flow cytometry data. So as you see, as you go down from that's without maturation and without sGP, this is with maturation and no sGP. And then as you add more and more sGP, you see this increased presence in the markers as they shift right. And one of these stood out the most to us, and this is this CCR7 when you add 1 microgram per milliliter of sGP. And so CCR7 is the marker that's present whenever a dendritic cell is starting to move to the lymph node so it can help fight this virus. And so there was this huge increase in the CCR7 marker present on these dendritic cells, indicating that they were moving throughout the body when sGP was added. So from this, we concluded that whenever you add sGP, even after maturation, they're enhanced. And so what do we find? We found that when you add sGP, there's enhanced expression of these CD83, CD86, CD54, and especially CCR7 markers on the dendritic cells. And IL-12 production is significantly increased whenever you go from just 1 sGP to 10 sGP. And so why would it be beneficial for a virus that's trying to infect and kill off all the cells to enhance this maturation for these dendritic cells? And it's because, as we saw in that CCR7 marker, which indicates movement, the virus has infected this dendritic cell and then matures it, and that dendritic cell starts moving all throughout the body to the lymph nodes. So it's basically getting transported free of cost all throughout the body, where it can infect and it's less likely to be caught and killed. And so from here, we would like to look at how the gene expression in dendritic cells differs whenever you do have sGP and whenever you don't have sGP. And these are our sources. And we'd like to thank Wofford College and Edward Via College of Osteopathic Medicine for providing funding and supplies for our project.